Pode sim.
Tá. Já aviso quando estiver gravando.
Estão vendo meus slides?
Sim, e tá tudo ok lá.
Pode começar?
Sim.
Então, good afternoon. This is the TEM 3, Human Health, from 4 p.m. to 6 p.m. I am trying to speak in English because one of our guests, which is Stuart, it's not a native in Portuguese language, so we are talking in English. This table, and we are 6 projects from the human health. I will briefly describe each of the 6 projects, and then I will describe the programmation of our table, and then I will pass to each one of our guests. Okay, so we are 6 projects. One of them is Healthy Aging and Urban Mobility, which PI is Professor Josimari from the Public Health Department. And we have 2 other programs participants, Physical Education and Architecture and Urbanism. The other project is Diagnosis and Intervention for a Healthy Lifestyle, which PI is Diego Silva, is here, Diego, led by Physical Education with the Public Health Program. And then we have Processing of Plant-Based Foods and its Health Impact, led by João Borges Laurindo, which is here also, I believe. Let's check if João is here with us. From Food Engineer with Food Science. Then we have Cellular Communication and Systems Biology from Edmundo Carlos Grisart, which is here also, I believe. Let's check. Is there. From Biotechnology and Biosciences with Neuroscience, Biochemistry, Physiology, and Pharmacology. Then we have Interprofessional Health Education, led by Jussara G. Martini, from Nursing with Public Health and Pharmacy. And finally, Molecular and Therapeutic Targets in Metabolic and Degenerative Diseases, from Ariane Pacheco de Souza, from Biochemistry with Neuroscience and Pharmacology. We planned 2 moments for this table in this evening. The first moment, we have 4 guests. Originally, we were 5 guests, but one of them, which is César Messias de Oliveira, he just sent me a message telling that he is in a place where there was a helicopter crash. Fortunately, not with him. He's very well and safe, but he is without electricity, and he is without power, and he is without internet. So unfortunately, César will not be with us. I am Eleonora Dorsi. Okay, I will talk very briefly for you about aging. And then our first speaker after me will be Andiara. Andiara is there. I saw her already there. Andiara, okay, you are here with us. From the Department of Kinesiology and Community Health from the University of Illinois at Urbana-Champaign in USA. She will have 10 minutes. She is from the project Diagnosis and Intervention for a Healthy Lifestyle, and the title of her presentation is Human Aging and Active Lifestyle. Then we will have our second guest, our second international guest, which is Stuart Phillips from the Department of Kinesiology, which from the project Food and Plant Origin, Processing and Impact on Health. He's talking about the impact of nutrition and exercise on the mechanisms of human skeletal muscle protein turnover. Then we have Tiago Fleming Outeiro from the University of Newcastle in UK, from the project Cellular Communication Systems Biology, and the title of his presentation is Collaborative Plasticity, We Cannot Do It Alone. And finally, our last guest is Marco Aurélio Alves from University of Porto. He's from the project Molecular and Therapeutic Targets in Metabolic and Degenerative Diseases, and he's talking about aging, metabolic diseases, and male fertility. The second moment of this table, with more 60 minutes, we will ask each one of the coordinators of the projects to talk briefly or to show the video, okay, about the activities of this project. And I will give now a very brief presentation of the people which are talking now. Starting by me, I am Eleonora Dorsi from the Department of Public Health from UFSC, Brazil. I am the principal investigator of the EPFloripa cohort study of aging in Florianópolis, South Brazil, and my research field is in epidemiology of aging and cognitive stimulation. César Messias de Oliveira, which unfortunately will not join us this afternoon. He is from the Department of Epidemiology and Public Health from University College London in UK. His main research field is epidemiology of aging, and he is one of the coordinators of the English longitudinal study of aging, the ELSA from UK, and also the Brazilian longitudinal study of aging, which is a similar study done in Brazil. Then we will have Angiara from the University of Illinois in Urbana-Champaign in USA. She is associate professor from the Department of Kinesiology, and she directs the Aging and Diversity Lab and teaches in the area of aging, human health, and global health. Her research informs public health policy and practice that can lead to healthier communities. Then we have Stuart Phillips from the University, McMaster University in Canada. His research focuses on the impact of nutrition and exercise on the mechanisms of human skeletal muscle protein turnover. He is keenly interested in diet and exercise-induced changes in body composition, particularly in older persons. Then we have Tiago Fleming Outeiro from Newcastle University in UK, which studies mainly Parkinson's diseases, Alzheimer's disease, alpha-synuclein, tau and amyloid beta. He is doing research on the molecular basis of protein misfolding diseases using a variety of model organisms from yeast to mice and rats. His team has developed key tools and assays for the study of protein interactions that are the root of protein aggregation, a key event in the neurodegeneration. And then we have Marco Alves from University of Porto. He is the lead of multidisciplinary research in biomedicine. He heads a group of research focusing on endocrinology, metabolism, and male reproduction. and he mainly works on male reproductive biology, metabolic modulation, obesity, and physiopathological causes of subfertility or infertility. His main line of research is devoted to the study of metabolic cooperation in spermatogenesis, metabolic-related infertility, and diabetes and obesity. Okay, so why I am here? First of all, I am officially not the coordinator of the Healthy Aging program, but Josimari, which is the official coordinator, asked me to be here. She is not, but I participated from the very, very early beginning of writing the call, which succeeded with this funding. And I'm very happy to be here sharing with the other colleagues from the other projects from human health here. And we choose because you can see by the subjects what is important about healthy aging. Life expectancy is approaching 8 years for most of the countries. And also in Brazil. Brazil is more or less 75 years, but here in Santa Catarina, we are already 8 years old living. And but this, which is a big achievement, is coming with a very high burden of diseases and limitations. And all of us, we are trying to get a healthier process on aging, which means from who, that everybody needs to be able to meet their basic needs, learn, grow, and make decisions, be mobile, build and maintain relationships, and contribute to society. So some of the targets are the environment, the public policies, education, physical activity, nutrition, and chronic diseases control. So we can see that the healthy aging is some of our key that bring all of us together. So we choose this subject to be the guideline of this presentation. Okay, so I was very brief, and I only wanted to welcome all of our guests and say that this is a very unique opportunity, that if it was not from this event called by PRINT-UFSC, probably these 4 people, our estimated international guests, which are here today, they would not meet, at least in a synchrony form. So let's enjoy this opportunity and make this the first of, I hope, other events like this for us to share and to collaborate and to improve the health and the healthy aging for all of us. Thank you very much. Now you can pass to Andiara, please.
Não sei se vocês conseguem me ouvir ou me ver. Está duplicada a imagem aqui para mim. Alguém pode me ajudar?
Estamos te vendo duplicada e estamos te ouvindo bem, Diara.
É? Ai, que bom. Eu não sei como fazer para não duplicar, mas eu me posiciono, então, do jeito que vocês conseguem me ver bem. E para eu compartilhar os meus slides, como que eu faço?
Você está vendo um maisinho no canto esquerdo inferior da sua tela?
Estou habilitando.
Ah, tá.
Agora está habilitado.
Eu clico no Upload a Presentation? Ou...
Isso.
Sim.
É.
E aí clique em enviar. Alexandre, você vai ajudar as pessoas com essa parte de carregar? Eu posso silenciar o meu microfone?
Sim, eu posso acompanhar. É que a pessoa tem que carregar, selecionar o arquivo e enviar.
Eu enviei acho que 2 vezes, não sei se vai dar algum problema.
Às vezes demora um pouquinho para carregar.
Ok.
Para ti voltou a tela com aparecendo as pessoas.
Oi, pessoal, voltei. Eu enviei meus slides para o Diego, não sei se, Diego, você poderia projetar eles. Eu posso mandar em PowerPoint também, se ajuda? Não sei, de alguma forma eu não estou conseguindo...
Alexandre, poderia me colocar como para projetar, para compartilhar?
Sim, acabei de lhe colocar.
Tá bem, obrigado.
Diego Silva, né?
Isso.
Ok, muito obrigado.
Isso, tá ótimo. Obrigada. Posso começar? Fica meio cortada a minha imagem, mas vamos lá. Ok, vou falar em português. Ótimo. Oi, pessoal. Vou falar em inglês, então, para que nossos colegas consigam entender. Thanks, Dr. Eleonora, for your introduction, and it's a pleasure to be part of this event and connect with my colleagues from Brazil, as well as to get to know new ones on the panel. As Dr. Dorsey said, I'm from the University of Illinois, and I will be talking to you about aging and physical activity. Can you, próximo slide, por favor? Okay. So as we know, Brazil is aging, and so is the rest of the world. The percentage and number of older adults is higher than ever before. Increases in life expectancy are a tremendous achievement in public health. But population aging, like Dr. Dorsey said, provides both challenges and opportunities. As researchers, policymakers, individuals, and society at large, aging is transforming how we approach health and well-being. We need to ensure that we are not only adding years to life, but also adding quality to the additional 30 years that has been added to the lives of Brazilians since 1940, as is represented in this graph. Next, please. So population aging is contributing to the epidemiological transition that we are seeing, where non-communicable diseases or chronic diseases such as heart disease, cancer, stroke, diabetes, COPD, and dementias, Alzheimer's disease is the most common, are increasingly becoming more common and affecting health and well-being of people at a greater extent. So the figure on your right side illustrates that currently the burden of disease in Brazil is attributed to 15 leading risk factors, including many health behaviors such as diet, smoking, and physical activity. So in the remaining of my presentation, I will focus on physical activity and describe the benefits, the public health recommendations, and talk about the 2 research studies that I've been involved in. So moving to the next slide already is, before I start talking about physical activity, what I talked to you in the previous 2 slides were general across all ages, and I want to underscore the case for older adults. The impact of non-communicable disease is even greater among those aged 60 and older. Non-communicable are by far the group of diseases and conditions that are causing most deaths and disability, causing more years of life lost than communicable, and injuries, as you can see in the blue bar on the figure. Can you go to the next, please? Yeah, so that's this slide that shows. So older adults are definitely a vulnerable group that is mostly affected, and that's why it's so relevant that we promote health among this group. Next slide, please. So let's just take a look at what are the benefits of physical activity. There is strong evidence to support that physical activity has a number of individual and societal benefits. Individual benefits include immediate and long-term, and a range, across various physiological, psychological, and social domains. So, for example, physical activity improves almost all aspects of cardiovascular function, helps regulate blood glucose levels, improves sleep quality and quantity. Resistance training can have a significant impact on the maintenance of independence in old age, reduce risk of fall and fall-related injuries. Physical activity can reduce stress and anxiety and can make important contribution to in the treatment of various mental illnesses, help postpone age-related cognitive declines. Participation in physical activity can help empower older adults and assist them in playing a more active role in society. And so society benefits too by promoting healthy aging and physically active older adults. So as physical activity can postpone the onset of disease and frailty, it helps reduce health and social costs. Physical activity improves the productivity of older adults. Many work as late in life. Promotes a more positive and active image of older persons, so fighting ageism and so on. So engaging in promoting physical activity is in the best interest of everyone, really. And if you want to read more about the benefits of physical activity, I suggest my own publication listed on the slide. Next, please. I want to pause, and this year, talking about health in 2020 is inevitable that we need to mention COVID. And I listed on this slide few articles illustrating the potential of physical activity being connected with less obvious health issues. So COVID complications and COVID mortality have been associated with pre-existing non-communicable comorbidities, all in which have potential association with physical fitness and physical activity levels. So I'm a great advocate for physical activity, and I want to argue that the benefits of physical activity transcends the typical realm of non-communicable disease. Next, please. So how much physical activity is needed for health benefits? Older adults should engage in 150 minutes per week of moderate intensity aerobic activity and resistance training twice a week. So the evidence is less strong for balance, but I want to also mention that balance exercise is recommended for all older adults as a way to prevent fall injuries. And I also want to note that the recommendation is not different for older adults than it is for adults, but it's important to note that if health conditions or maybe other factors prevent activity at recommended level, older adults should perform physical activities as tolerated so as to avoid being sedentary. Avoiding sedentary behavior is an important consideration when promoting health and well-being among older adults. Next, please. So a logical next question is to understand how many older adults actually meet the recommendation and are physically active. The answer is the prevalence varies by studies. based on how physical activity is assessed, age of participants, and many more factors. In general, less than a third of older adults in the US report meeting recommended physical activity guidelines. Some studies in Brazil showed prevalence ranging between 30 to 60%. So there's kind of disagreement sometimes in the literature, but studies agree overall that older adults are one of the least active populations. And I think it's important to note that some subgroups within older adults are also affected, and disproportionately even affected, including the oldest old population, women, older adults with limited educational attainment, those from low socioeconomic status, ethnic minority groups, and rural dwellers. Next, please. So understanding factors that influence physical activity behavior has been a topic of research to many scientists, including some of our very own panelists here. And I like to use the socio-ecological framework to understand the complex layers of barriers to physical activity and program participation. So in the bottom, we can see that interpersonal factors that include individual factors like physical and mental health. These are important factors that might present significant barriers to physical promotion. So for example, older adults that go through disease, that have disease and need to go under treatment, they are experiencing pain or have fear of falling. These are all important factors that influence the participation of older adults and my limited and engaging in more active lifestyles, limited health literacy, lack of motivation, and preference for sedentary behavior. So these are all individual factors that are important to note when we want to promote physical activity. And then the ecological model goes into higher levels that include interpersonal factors that are based on interactions with other people and with the community. And then other factors like environmental, structural, and organizational factors. These are all extremely important factors that to me underscore the shared responsibility that is needed to change the scenario of physical inactivity among older adults. For example, how can we ask older adults to walk more when the community where they live in is unsafe, or has uneven sidewalks for older adults to be active and risk them to fall, increase their risk for falling? How can we ask older adults to meet the physical activity guidelines if, for example, the option presents a cost and is too expensive for older adults to use a facility or even drive to the facility or take the transportation to that? So there are interesting factors there that we need to take into consideration. Next, please. So thinking about these barriers and possible solutions for community-based programs that could have long-term benefits, be sustainable, and have low implementation costs, my colleagues and I started a conversation about possible partnerships with the SUS, the Brazilian National Unified Health System, to expand the work that they do within the family health strategy, or the Programa de Saúde da Família. Can I have the next slide, please? So that's when we developed Vamos, which is a behavior change program that strives to motivate older adults to engage in physical activity and healthy eating. This is a partnership among UFSC, led by Dr. Benedetti and other researchers from Illinois, Nebraska, and the city of Florianopolis. So the program is offered at community health centers, or the Unidade Básica de Saúde, and we train their own staff to deliver the 18 sessions that last around 1.5 hours to 2 hours during a 9-month period. Findings are encouraging in many ways. It's shown to be effective in changing people's behavior. The centers and their employees were able to adopt the program. And despite some limitations and issues that we need to address, our partnership with the city of Florianopolis is only expanding as we speak. So another community program that I've been involved with also built on the SUS and the family health strategy, now in the southernmost state of Brazil, Rio Grande do Sul, in a rural town that names the study Dona Francisca. So this is a partnership among Illinois, the Federal University of Pelotas, UNESP, and the city, UNIFESP, and the city of Dona Francisca. So this is, again, a behavioral change program that strives to motivate older adults to engage in physical activity, healthy eating, and also adds an element of emotional well-being through mindfulness techniques. In this program, we train community health agents to deliver the 6-month-long program to older adults in the community. We also wanted to increase motivation, and the study employs religious and spiritual content throughout the program. Findings are promising and show the real possibility of leveraging existing resources to change the culture of health among older adults. So this is my last slide, and I want to circle back to the beginning of my presentation, sharing with you this quote. The power of community to create health is far greater than any physician, clinic, or hospital. So in my opinion, part of the solution to improve health and well-being among older adults lies not only in developing state-of-the-art care and treatment, but also by addressing underlying factors that influence everyday lives of individuals and the important choices they make. So that's all I have for you. Thank you for listening.
Thank you very much, Indiara. We have now 5 minutes to question.
I think they're going to show me. I saw in the images. I don't see anything anymore.
Quem será o próximo a apresentar?
Nada, Leonor, ainda.
Coloquei o Stuart como apresentador.
See my screen?
Yes, Stuart.
Yes, yes,
Okay.
you can see.
Well, thank you very much for the invitation to speak. Bom dia a todos. Portuguese is not so good, so this is all in English. My apologies. I hope that everybody in Florianopolis, which is a beautiful part of Brazil, are all safe, and everybody is doing okay amidst COVID. These are the people responsible for the work that my lab has done over the last probably 5 to 10 years, and I'm going to highlight some of them and their particular contributions throughout this presentation. I'm going to talk to you today about muscle, and particularly muscle in older people, and convince you, hopefully, that muscle is important. One of the key statistics that I like to discuss is that sarcopenia, or the age-related loss of muscle mass, is very prevalent. Probably fully one quarter of older people over the age of 65 have sarcopenic syndrome by various definitions, including a loss of muscle mass and a loss of muscle function. And that can be as high as up to 60% of people who are over the age of 80 years. And the key point to make is that as muscle mass declines, so does functional capacity. And the area of blue shown in this slide here is an area which, when people decline below this, their risk for functional disability increases. So clearly, muscle matters. One of the first things I'm going to make, and one of the points, is that older people are thought to require more protein than the minimum RDA. The recommended dietary allowance, or RDA, sits at 0.8 grams of protein per kilogram body weight per day. And recommendation have been that the minimum protein intake for healthy people is probably in the range of 1 to 1.2 grams per kilogram per day, probably even higher in situations of acute or chronic disease, and definitely in situations of severe illness or injury or malnutrition. It's advisable to go up to intakes that are as high as 2.0 grams of protein per kilogram body weight per day. And the references are given right here. So I want to talk to you about how muscle mass is regulated. And the easiest analogy I can use is that it's like a brick wall. There are bricks coming in at one end, and there are bricks being taken out at the other. The bricks coming in are obviously the amino acids that are building up muscle protein, and the bricks being pulled out are damaged, if you like, amino acids. So you may have seen these types of curves before, and so my apologies if this is something you've seen before. But the two processes we're talking about are muscle protein synthesis, or bricks going into the wall, and muscle protein breakdown, or bricks coming out of the wall. When we wake up in the morning, we have a marked stimulation of protein synthesis, a suppression of protein breakdown, and the area of green then is a small amount of muscle being gained, or the wall is getting bigger. But when we don't eat for a period of time, the rate at which the bricks are going in declines quite sharply. The rate at which the bricks are going out increases a little bit, and you can see we have this gray area of muscle protein loss. But then we eat our lunch, we get another stimulation, we go for a period of time until we eat our dinner, and we get another stimulation. What exercise does in this scenario is it amplifies all of the periods in which you're experiencing a gain in muscle mass. So the addition of exercise enhances the muscle's sensitivity to the provision of protein. And I'll show you what that looks like. So each time a person, after they have performed resistance exercise, consumes protein, they get a little bit more muscle growth. In the fasted state, they lose a little bit less muscle. Over time, you can see that this pattern would result in muscle being gained, or in an older person, less muscle being lost. In fact, one of the phenomena that we have been able to document, and a number of other groups have also corroborated this finding, is that in older people, there is a phenomenon known as anabolic resistance. And that describes the phenomenon in which each fasting state gain fed state gain in muscle protein balance is actually less. And so over time, an older person doesn't achieve the same robust stimulation of muscle protein synthesis, and so their muscle begins to decline. We can recreate this scenario in an older person, or in a younger person, by putting them on bed rest. So physical inactivity recreates very, very faithfully the process that you're seeing here in older people, which is anabolic resistance. In fact, we've been able to quantify on a per meal basis how much protein is required in an older person to overcome this phenomenon of anabolic resistance. These are a group of younger people on the left and older people on the right. And we're looking at the rate of muscle protein synthesis increasing and then the rate at which it plateaus. And you can see the inflection point in the curve here is about 1/4 gram of protein per kilo. Whereas in older people, it's almost as high as 0.4 grams per kilo, or about 70% higher, but to achieve the same protein synthetic response. So older people can get to the same response, but they need more protein to do that. What does that really look like in real terms? This is a diagram drawn for me by a former PhD student, Sierra Oikawa, and she's shown here we have a small amount of protein at breakfast, a larger amount at the lunchtime meal, and maybe the largest amount at the dinner time meal. And she's also shown you a bedtime meal, pre-sleep meal here as well. Well, if you're a younger person weighing about 85 kilograms, that dose of protein at 20 grams here, you hit the threshold to stimulate muscle protein synthesis twice in one day. If, however, you're an older individual and you require 0.4 grams of protein, you only hit this threshold to maximally stimulate muscle protein synthesis once a day. And it really brings into question the source of the protein and how important that is. One of the key complications that in aging is a period of what we call a disuse period. And the usual anabolic resistance in somebody who is maintaining their physical activity, it would certainly be downward in direction, but an older person who would experience a period of disuse would experience an accelerated period of muscle protein loss. This could be hospitalization, surgery, or even a period of inactivity as a result, for example, like the global pandemic that all of us are experiencing right now. And so Sarah drew this sort of cycle where an older person experiences this anabolic resistance during a disuse event, surgery, illness, or bed rest. They experience a decline in protein intake and energy intake, and then they get a dampened muscle protein synthetic response. They lose muscle. As a result, they're doing less, and this becomes somewhat of a vicious cycle. So what can we do in older people to intervene in this cycle and try and overcome the anabolic resistance of disuse? One of the approaches has been to give older people a protein supplement, and this is work from Doug Padden-Jones, in which younger individuals were on bed rest for 28 days, and an experimental group got a high dose of amino acids during the bed rest period, and you can see that their change in leg lean mass or leg muscle mass was prevented, but not in the control group. Interestingly enough, at the same time, this is the change in strength, and you can see that the preservation of muscle was associated with a preservation of strength. In other words, the group receiving the amino acid and protein supplement were able to offset these losses. We think that this is particularly important because, as opposed to younger people who can reclaim their lost muscle, older people, sometimes a period of disuse, for example, can represent a period of muscle loss which might be permanent. This is another study from Kirk English working with Doug Padden-Jones again, and you can see this is a control group and this is a leucine supplemented group. They got leucine with every meal. And this is the change in whole body lean mass, and leg lean mass. And you can see that from pre to post, that the decline in the control group was always greater than the decline seen in the leucine supplemented group, which really does point a finger to the key amino acid in this whole equation, which is leucine. It's one of the 20 essential amino acids. Short-term bed rest-induced atrophy can be offset with leucine-rich protein supplementation. So some take-home points, what I've told you. First of all, protein requirements in older persons appear to be elevated, and many older people don't consume protein at the levels required to meet the estimates of greater protein need. At the same time, the amino acid of all 20 that appears to be particularly important is the key amino acid, which emphasizes an important role of protein quality in delivering nutrition to older people, and dairy proteins would be one of the most effective ways of delivering that source, and not the least of which dairy proteins in the form of foods would also contain a number of other nutrients that would be important to improve diet quality in older people as well. And so aggressive nutritional support, particularly in periods where older people are unable to be mobile, will be important to slow down losses of muscle mass and support recovery. We clearly need some longer term trials with larger numbers to emphasize the importance of these points and their ability to be able to be mitigated. So more research, longer term trials are clearly needed. With that, I'd like to say thank you very much for your time and your attention, and I look forward to hearing from the rest of the presenters.
May I ask you something, Stuart?
Oh, yes, please.
Oh, thank you. First of all, thank you so much for your fantastic presentation. I was wondering if you could talk a little bit, very briefly, about how to get leucine from diet, because I don't know if this is feasible to get supplements. I am always thinking from a public health perspective, from a public health policy perspective. What do you think about that? Please.
Yeah, so the advice I would give somebody is always food first, and then if it's not able to be done with food, then supplements are really the next best choice. The good news is the highest quality proteins that people can consume that also have the highest amount of leucine or dairy proteins. So milk, cheese, yogurt would be particularly good sources. Eggs are also good sources, mostly animal source proteins, to be honest. But to be really clear about the two sort of potency of the stimuli that are out there, the more active you are, the better the leucine works. So it's physical activity on top of a high quality protein containing diet, but definitely dairy source proteins. But your point about supplements versus food is well taken. Not everybody can afford supplements, but I would try and emphasize dairy source proteins. There are an interesting number of plant proteins that are now coming out to be shown to be actually pretty high in leucine.
Thank you. So your presentation is very in line with Andiara's. More questions, comments?
Stuart, can you hear me?
Yes.
Oh, thanks for your talk. This is Edmund from UFSC. So, sorry if I have missed, but what are your thoughts about the protein degradation and absorption in the light of the human microbiome, the different microbiomes we have, so the way we deal with the food and digestion, it's individual. You know?
Yeah.
So what are your thoughts about that?
Yeah, it's a good question. I don't think that there's much doubt that as people get older, probably the efficiency of absorption of protein goes down. So that's probably part of the reason why older people need more protein to get back to where they were before. The question about the microbiome is a really good one, and I'm afraid I don't have a good answer for you. The hospital where my university is deals in gastroenterology, so I listen to lots of presentations about the gut microbiome, and I think it's going to become a pretty important player in understanding why some people do better with certain interventions, even exercise, and other people don't. But I think we're a long way from understanding what happens in people than we are from understanding what happens in models of mice. So in the mice and the mouse models, it seems they've got a lot of these things figured out, but certainly it'll be important in the future to take the gut microbiome into consideration for sure.
Thanks a lot.
My pleasure. All right, looks like we're ready to move on. Thank you.
Thank you, Stuart.
Tiago?
Oi, boa tarde.
Oi, querido, pode começar.
Obrigado. Então, boa tarde a todos. É um prazer estar aqui desde da Alemanha, não de Newcastle, mas da Alemanha, falar para todos vocês. Eu sei que é suposto ser em inglês, portanto vou passar para inglês. So I picked this title, Collaborative Plasticity, because I think one of the important points of this program is really to highlight the importance of collaborations. And this stems from the fact that, of course, science is so specific these days that we cannot solve problems alone. And programs like this one that you have, I think, are really essential for enabling us to advance faster and further in our quest to understand, in this case, human health. So I'm talking to you from Göttingen, this little university town here in the middle of Germany. Perhaps you've never been here. The closest larger city is called Hanover, and this is also not too far from Frankfurt. And Göttingen is a nice little university city with lots of interesting sites to visit, famous sites like this observatory from where Gauss did a lot of his work, looking at the skies. And then there's also some important landmarks in the city that you're welcome to visit if you have a chance. So this slide summarizes the interests of my research over the past over 20 years now. We've been interested in understanding what happens to proteins during their normal lifetime that allows them to fold and to adopt a three-dimensional structure that allows them to perform the functions they are supposed to perform. And we're interested in studying this process also from the other perspective, meaning we know that some of these proteins sometimes also, for reasons we don't fully understand, end up misfolding and accumulating, and we know this can be associated with disease. And to tackle this, we need to understand what happens inside a cell. And here you see a typical textbook picture of what a cell, what we think a cell looks like. And you see all the different organelles and the cytosol and ribosomes, these little dots. But the problem is that it's really hard to capture the actual environment of a living cell in a two-dimensional picture like this. So in fact, we know that inside a living cell, the environment is extremely crowded. There's a lot of proteins, like we heard from the previous speakers. Stuart told us about proteins, and we know the protein concentration is really high inside a living cell. And so we're interested in studying how cells respond to the accumulation of misfolded and damaged proteins, and also in understanding what are the consequences of protein misfolding inside the cell. So what happens is that if cells cannot respond properly, then you can have problems. You can have different types of diseases like those represented here, of which Alzheimer's is by far the most common disorder, followed by Parkinson's disease, and then several others are less common, and some are studied by colleagues at UFSC, like Professor Patricia Brocardo. She studies Huntington's disease, and we'll talk a bit about this maybe during the questions at the end. But we study these diseases because we know that they are all associated with the deposition of different proteins in different parts of the brain. So Parkinson's disease is one of the most common, as I just mentioned, and it's typically known for the loss of these dark pigmented neurons from the substantia nigra called dopaminergic neurons. And we know that in the areas in the brain that survive, we can find the presence of Lewy bodies that we think are made of fibrils like this one shown here that are composed of a protein called alpha-synuclein. So this protein is really central in the disease. But we need to keep in mind that Parkinson's is also not just a disease of the substantia nigra. It affects a lot of other areas in the brain, such as the cortex and the brainstem, hippocampus, and many other areas as disease progresses. So this is important for the studies we will talk about a bit further down the road. So we don't understand why some of us end up developing diseases and others don't develop, unfortunately. And of course, we know that we are all different. But genetics doesn't explain the majority of the cases. It actually explains only 5 to 10% of cases of Parkinson's, and this applies also in the case of Alzheimer's or ALS. It doesn't apply in Huntington's disease because that's a purely genetic disorder, but in Parkinson's, genetics, and we now know a lot of different genes associated with familial forms of the disease, explains only 5 to 10% of the cases. We study this protein a lot, alpha-synuclein, that I mentioned earlier, but we study other genes associated with the disease as well. So alpha-synuclein is a small protein that we are starting to understand to some extent, but it's also in many ways a protein we don't fully understand in the context of certain cells, like in the blood, in red blood cells. We don't know what the protein is doing there. So there's a lot of open questions, and we are interested in understanding how this protein really relates to disease and to Parkinson's disease. And one of the emerging concepts in recent years has been this idea that pathology in Parkinson's disease spreads throughout the brain as disease progresses, and it goes from the brainstem upward, it goes through the substantia nigra where it damages the dopaminergic neurons, and then eventually goes further up into higher areas of the brain, including the hippocampus and the cortex. So this is really just to highlight the importance of studying the whole brain, not just the substantia nigra. And what we try to do in my research at the molecular level is trying to find Wally, or understanding how cells, how neuronal cells find Wally, find the damaged proteins. And of course here, just so you don't stay curious, it's there, our little Wally. And that's what cells need to do so that they can take care of this protein. But if we think about the functional implications of the accumulation of misfolded proteins, then we can focus on the hippocampus, which is a region I'm sure a lot of you have studied a lot in detail. And this is a region that we've been interested in because of the possibility for doing functional readouts and for measuring synaptic plasticity. And the hippocampus is also a region that Professor Patricia Brocardo has been interested in the context of plasticity in the brain. So some years ago, we used this LTP paradigm to study the effect of alpha-synuclein in the brain. We made discoveries. And then a few years ago, we connected with Professor Patricia Brocardo, and we talked about our findings, and we wanted to extend this and to start collaborating. So this actually happened in a visit I made to São Paulo. That's where we met. And since then, we've been in touch, and we've actually exchanged students. Evelini was our first student, and hopefully next year, Patricia will visit us in Germany. So this is really a tripartite collaboration between Florianopolis, Newcastle, where I also have an affiliation, and Germany, where I'm located most of the times. So this is something to train our spatial plasticity, if you will. And I just want to highlight some of the studies that we started doing with Evelini when she visited my lab in Newcastle. So she was studying adult neurogenesis in the hippocampus. And the reason we could do this is because in Newcastle, we have a very good human brain bank from people that had a variety of different diseases. So Evelini joined us, and she was using the brain tissue in the brain bank in Newcastle to look at human adult hippocampal neurogenesis. This is based, of course, on the fact that there had been already previous studies in Parkinson's disease showing that perhaps neurogenesis is affected in patients. And here we could expand the study by looking at a larger number of samples. We looked in this initial study at 10 control and 10 Parkinson's patients using standard immunohistochemistry and immunofluorescence stainings. These are just some of the cases. We're lucky to have access to patients with a wide variety of ages, from 60s, here 48, to actually over 100 years old, which was quite impressive. And I'll show you just an example of some of the data Evelini collected after standardizing all the protocols for staining for the correct markers, double portin. Here shown in red, this was one of the markers of neurogenesis. And after a lot of work and a lot of imaging, Evelini was able to take a lot of nice pictures that she was able to reconstruct here of the hippocampus, showing the marker of neurogenesis. And just to summarize, we found something really interesting, that we could label neuroblasts even in a very old individual, or maybe experienced individual with over 100 years old. So this was really interesting, and we are now trying to look at this in more detail, and that will be the goal of the next steps of the project, also in combination with the animal models that we have here in my lab. So this is important because in all these neurodegenerative diseases, we can detect the clinical symptoms, and for some of them, like Parkinson's, we have certain therapies that actually work pretty well, but we don't really know the causes, and this is where we need to invest a lot, so that hopefully in the future we'll be able to have better therapies. So I stop here. I would like, of course, to thank Evelini and Patricia for the collaboration, which has been great. Also a postdoc in Newcastle that has been very helpful in supervising Evelini, and of course to you for your attention, and the CAPES PRINT program for supporting this initiative. So thank you very much.
Thank you, Thiago. Wonderful presentation. Can you hear me?
Yes.
Oh, great. Now, can I ask you something? I think we have 5 minutes or something like that. I was delighted by your presentation, and I wanted to ask you, because I work a lot of cognitive stimulation for older adults, and especially digital inclusion and cognitive stimulation done via devices, technology devices, and we see many of them improves a lot, and we don't know the pathophysiological behind this. We really don't know, because I wanted to ask you, maybe for Parkinson is easier, because Parkinson has a pathophysiological basis more defined, more clear than, for example, dementia, or from Alzheimer, for example, where you cannot find a really good connection between the proteins and the problems in the brain and the cognitive performance of the person. How do you feel about that? Is it possible that with stimulation the brain gets new connections and surpasses all the organic damage? How do you think about that?
Yeah, that's a very interesting question. So actually in Parkinson's, we know that deep brain stimulation, this surgery where you insert some electrodes in some important nuclei in the brain, you can stimulate them and actually compensate for a lot of the motor deficits these patients have. So this does not work in all patients, and we're still learning as we go along which are the nuclei that we could stimulate. But this actually fits with your question, and this is an example that it's possible to stimulate the brain in ways to compensate the lack of cells and of stimulation that happens due to cell loss. So there are now a lot of people trying this also in dementia and in other diseases like Huntington's disease, and there are promising results already. But of course, as you said, it's more difficult in dementia because the cell loss is so much greater that it's hard to stimulate areas that would compensate for the dementia. Now, what is also important is to make efforts towards early diagnosis, because it's estimated that by the time these patients are diagnosed, a lot of the cells in the brain have already been lost, and so it's important to diagnose them earlier on, so that perhaps strategies like the one you're proposing, stimulation, or other hopefully pharmacological approaches, may be able to intervene at that point and stop disease progression. So this is fascinating. There's a lot to be done, and hopefully we'll make some progress through collaborations with colleagues like you.
Thank you very much. Any more comments or questions?
Tiago,
Sim?
you hear me?
Yes?
Oh, this is Edmundo. How are you?
Hi.
Nice to meet you in person. My mom has Alzheimer's disease, and she was diagnosed at the age of 82, so it was quite late. And what is your perspective, your thoughts about mixing what you said? Lovely job, lovely work. I like it a lot. But what are your thoughts about aging at her age and this kind of hippocampal involvement in that? So aging is a difficulty factor to understand this kind of protein you're studying. So what's the relationship between these two systems, all the systems you're working on, and aging of the hair age or elderly age?
Oh, that's a wonderful question. Of course, we know aging is the major risk factor for the onset of pretty much all of these neurodegenerative diseases. We know that the risk increases tremendously. So from for Alzheimer's disease, from a risk of around 3% at age 65, it goes up to close to 40% at age 85. So this really shows that there is a strong connection. We don't know the molecular connection yet. Of course, in the aging field, there's a lot of exciting discoveries in terms of epigenetics, in terms of pathways, signaling pathways that we know are important, in terms of enzymes that we know that at least in model organisms can be modulated in order to delay aging. But I think it's also important to keep in mind what we can learn from humans and from this centenarians that live well into their hundreds, and well, after 100 years old, I don't mean more than 1 century, of course, but we know that they do not necessarily develop dementia. So this is telling us that there is something in biology that we should learn about that maybe enables us to delay or prevent cognitive decline. Of course, it's still an area we need to do a lot more, but for sure, I think studying aging will be invaluable for understanding how to treat and how to prevent these types of diseases in the future.
Thank you. Thank you for sharing your knowledge, and thank you for joining us on this workshop.
A pleasure. Thank you.
Can I move on to the next? Thank you so much, Thiago, and then now Marcos Alves.
Hello, are you hearing me? Hello. Yes, Marco.
Yes.
Okay, so thank you very much. First of all, thank you for this kind invitation to be here to talk a little bit about our research and how CAPES has been helping us to do this collaboration. I will start to show you some of the subjects we work on and that we have some ongoing collaborations with the support of CAPES. And then I will also like to show you some of the projects and some of our facilities that can potentiate, I hope, some of other collaborations with you at your institutions. So I will, okay, I will start to talk about the team and network, and then a little bit about aging, metabolic diseases, and fertility, and how it is a public health problem, the infrastructure and collaborative possibilities, and also ongoing work and future perspectives on the subject. First of all, who are we? We are Sertoli Cell and Gamete Biology Lab. We work in a multidisciplinary team with specialists in male reproductive biology. We are mostly biochemists and also physicians. This is our group, which is very extended, as you can see, but we are always keen to receive new students from abroad to help us to improve some of these works that we have ongoing here. So we are part of a large network, a national network from University of Aveiro, Lisbon, and also Coimbra and Porto, of course, together with assistive reproductive services and fertility clinics all over Portugal, and also abroad. From abroad, I can tell we collaborate with UCL in London, in Finland, we have collaborations in France, in China, India, and now in Brazil also, as well as in the United States at the Rockefeller University and also at the American Center for Reproductive Medicine. So we are part of a national network that has expanded to an international point of view. And of course, CAPES has helped us to implement these collaborations in Brazil, also in Florianópolis. So if you look at the evolution of fertility rates worldwide, you can see here in the 70s and here in 2014, and of course the colors where you have many children in red and purple, and few children in blue. You can see that from the 70s to 2014, even Africa has lowered the number of children that the couples have. So this means that the fertility rates worldwide are decreasing a lot, even in countries that we would not expect that to occur. So in male fertility, there is a huge problem. Very recently, there was a systematic review showing that from the 70s until 2010, there was a 52.4% decline in sperm concentration and a 59.3% decline in total sperm counts. This means that male fertility is highly decreasing in the last few decades. If you look at the numbers, sperm concentration in the 70s was expected to be like 92.8 million per milliliter and has decreased in 2011 to 66.4 million per milliliter. Total sperm counts are also decreasing a lot from the 70s to 195 million spermatozoa to 212 million spermatozoa. And to accompany this highly decrease, what we have from the World Health Organization is lower standards in sperm quality. What do I mean about this? 10 years ago, to have a normal fertility, the man was expected to have a higher concentration and a higher count of spermatozoa than nowadays. So instead of correcting and studying the problem and see how we can change it, we are just correcting the thresholds lower. So this means that we have here an health problem to account. I'm sorry. Also important is that we talk about delaying parenthood in women, and it is of course a huge problem because after 40, women will have problems to get pregnancy and to give birth. So this is very relevant. But if you look to the trends in the father, it also accompanies that. So this means that the father has also delaying parenthood later in life. And if you see, even in a few years here, just from 96 to 2010, you see that the age of the father is changing a lot, so in the last few years. And some studies have shown that the increased age of the father is also associated with some problems in the health of the children. For instance, autism has been shown to appear when the father is older. So it is important to establish a threshold or an age or something that can be useful as a biomarker for the father. What is the ideal paternal age? This is something that no one knows, but what we know is that aging, it causes decreases in sperm parameters, in sperm production, in viability, in motility, in volume. Also hormonal alterations, very important, with a high decrease of free testosterone. And you know that there are some men that take testosterone later in life. There are offspring diseases like genetic disorders, mental illnesses, and congenital abnormalities that can be related with a higher age in the father, and also more time to pregnancy, higher abortion rates, lower pregnancy rates are associated with a higher paternal age. In addition to that, there are also morphological and biochemical alterations and structural alterations even in the testes, with a decreased testicular volume, with a decreased testicular vascularization, and also problems with the functioning of Sertoli cells that I'm not going to talk today a lot, but is something that we work a lot, which they are the somatic cells of the testes that are responsible for the blood-testis barrier and also for the nutritional support of spermatogenesis. So it is very important to determine what is ideal paternal age. Is there a problem when there is an older father, and can we avoid those problems? So this is something we should be careful. So, but also important with age, as we have heard this afternoon, and you also know, it's the metabolic access, because with aging, people tend to have more metabolic syndromes, problems, metabolic diseases, diabetes, also obesity, and a decreased metabolic rates. So in 2017, we reviewed what happened with obesity, energy balances, and the impact for spermatogenesis, and you can see that there is mechanical problems that arise from obesity, like erectile dysfunction, that of course will have a higher impact in the sexual health of the individual. And of course, that obesity also causes high differences in hormonal levels, LDH, FSH, and also testosterone are highly decreased with obesity, something that is somehow common to aging, but we'll talk about that later. It can also cause a higher permeability of the blood-testis barrier, and this is important because if you have higher permeability in the blood-testis barrier, you will have things passing that should not be passing. So this means that spermatogenesis can be arrested. From the evaluation of the endpoint, and I would like to clearly state this, that sperm quality is only the endpoint of spermatogenesis, so you cannot say everything about the reproductive health looking only to the number of spermatozoa. You have to see the quality of the things. So you can have no changes in morphology, but you have lower motility and lower concentration caused by obesity. In addition to that, you all know that if you have increased beta oxidation, you will end up having more reactive oxygen species and more oxidative stress. But probably those that don't work in reproduction don't know, but spermatozoa don't have antioxidant defenses. So this means that if you have more oxidative stress than you should, they will be killed or will be destroyed by some mechanisms. So if you have alterations in obesity and energy balance, of course, you can end up by having compromised sperm quality and compromised fertility. Sorry. Okay. Okay, are you hearing me well? Because something lost here. Are you hearing me?
Yes, Marco.
Okay. Okay. So how do we approach these topics, and how you can also collaborate with us beyond what we already have with Florianópolis University? We have usually an untargeted metabolomics approach where you have a sample. It can be a liquid, it can be a biopsy, it can be a tissue, it can be cells that we can extract the metabolites and then analyze it. We do a target metabolites, metabolomics. Then we have a metabolite annotation where you can determine all the metabolites there are there, or at least most of them. We find metabolite candidates. We study protein, gene expression and data, and we can analyze the pathways that are involved and at the moment, or even do kinetics if it's possible, to determine the affected pathways by the diseases. We have cell-based screen analysis and also chemical biology screen. And we do in vivo studies with animals, and sometimes, and most of the times also with humans, with those clinics that I presented in the beginning. So this is how we approach that. Regarding our immediate facilities, we have, of course, an NMR-based metabolomics high profile, which means that we do a lot of metabolomics in cell lines, of course, in human populations, and also in animal models. And here we collect samples. We can build metabolic maps and try to unravel which metabolic pathways are affected by determinant diseases, in this case, aging, and also by metabolic diseases like obesity or diabetes. Very recently, only this year, we received a seahorse equipment which allows us to study high-risk spirometry and do the mitochondrial profiling in the living cells and living tissues, and biopsies also, by a plate. So this is something that we are very interested in start. So if you are interested to use this, please contact me and we can establish collaborations. We have ongoing three lines of collaborations where we can work on very much. One of this is obesity, which is a project that aims to work with obesity and fertility and see how these two issues can be connected and by somehow determinant for the health of the patients. Also this one called Traitor, because here we want to determine which information the spermatozoa carries to the next generation, so to the sons, regarding both aging and metabolic diseases. And we also have one line of research which is applied to sperm preservation and gametes, not only sperm but gametes preservation, by creating new media to do this gametes preservation for reproductive technologies applications. We are interested to show how aging affects male fertility and the reproductive outcomes of assisted reproductive technologies. Probably those that don't work with in vitro fertilization and with reproduction may not know this number, but in vitro fertilization can cost up to from 50,000 euros to €500,000 to the national health system and also in private clinics. So this is a very expensive world, I would say. Which molecular mechanisms mediate aging metabolic diseases impacting male fertility? So we have two major diseases affecting nowadays, and we need to know which are contributing to each and how is this correlating with this pandemic in also in the decrease of male fertility that I've showed you, with 60% decline in sperm counts in the last decades. And it's very interesting and not a coincidence that this is happening at the same time we have the pandemics of obesity and diabetes and other metabolic diseases. So perhaps there is here a strong correlation between these things. And we also want to know if there is an inter and transgenerational effects of aging and metabolic diseases that are passed by spermatozoa. This is very important because for many years, people would say that spermatozoa is not important and it does not pass much information because our site is much bigger and spermatozoa is transcriptally silent, so this is not important. This was not important, but the things have changed a little bit, and nowadays we know that the information that the spermatozoa carries is also pivotal and determinant to the next generation. So it is important to know what is there and what is the information that is passing. Regarding these two worlds of metabolic diseases and aging, there is a lot to do because not much has been done in the last few years. So I would like to thank you all for this, to acknowledge CAPES for the invitation and also for the collaboration that has been done in the last year with some labs here at the biochemistry department. And I hope that you can collaborate in other research projects that you have and be interested to speak with us. So thank you very much.
Thank you, Marco. Fantastic presentation. Very, very interesting. I don't know, but I would like to share with you that we are working in the project about healthy aging and urban mobility with lots of cohort studies, and many of them in Florianopolis, in Brazil, and in UK. In some of them, almost all of them, we have biologic bank. So maybe, I don't know if you can think about any collaboration using the data from these cohorts for your study. I don't know, it occurred to me now.
Yes, we have a large experience now in the last, I don't know, 2-3 years. We have implemented the protocol to analyze human plasma. So we can, if you have plasma, it's very easy to establish the metabolic profile of your cohort, for instance, if it's humans, and related with some of the data you have from the medical aspects. So this means that the problem with human cohorts is that you needed to have it very well established, and all the medical information. If you have that, and if you have some biological samples, urine is not that easy. Urine is not that good to work, but the blood, plasma, it's very easy to establish a metabolic profile.
Great, we do have that. We can talk more later because now we are ahead of time, but thank you very much. I pass to Diego that will lead the second moment of this presentation. Is that okay for you?
Yes, Leonora.
Thank you, Marco.
Now yes. Hello everyone. The second half of this table will be the presentation of the all coordinators of the human health field. We have a few minutes for presentation of each one of the coordinators, and we have a sequence for this for sure. I'm Diego Silva. I'm associate professor in the Department of Physical Education at UFSC. And now I pleasure to introduce the coordinators of the project. The first one will be Professor Eleonora Dorsey of the Graduate Program in Public Health. The second one will be myself. In the sequence, we have the presentation of the Professor João Borges Laurindo of the Graduate Program on Food Engineer. In the sequence, we have the presentation of the Professor Edmundo Grisart, who is presenting the graduate program on biotechnology and biosciences. After that, we have the presentation of the Professor Ariane de Souza, presenting the graduate program on biochemistry. And finally, we have the presentation of the Professor Jussara Martin. So, we have presentation. Eleonora Doss will be the first one. Alexandre, a primeira vai ser a Leonora. Professora Leonora.
Thank you, Diego. As Diego told, we are starting now to talk a little bit about each project. I am with Josimari working in the healthy aging and urban mobility project, which is led by the public health program with partnership with physical education and architecture and urbanism. And the main idea behind this project is that, of course, as has been told here, physical education, physical activity, nutrition are all key aspects of healthy aging, but this is not an individual subject. This is much determined by the environment. So we are together with the urbanism and architecture and the physical education program trying to study some of these key aspects. And we have a lovely video that was done by all the team. It's it's a big team. It's a wonderful team. We've been working very hard the past year, and we made a lot of big achievements. And now I would like to show you the video prepared by Renato Saboia. Thank you very much, Renato, for this work. So if you can pass the video now, Alexandre.
Qual dos vídeos? O primeiro aquele, né?
Aquele do Renato. Ele mandou um link novo agora há pouco. Renato Saboia. Se eu clicar nele, ele abre?
Não, tem que carregar do link. Eu vou pegar ali das notas partilhadas, eu peguei a última versão que ele mandou.
Obrigada.
Ok.
Olá, sou Josimari Telino de Lacerda, professora do Departamento de Saúde Pública e do Programa de Pós-Graduação em Saúde Coletiva da UFSC. Coordeno o projeto Envelhecimento Saudável e Mobilidade Urbana no âmbito do PRINT. Infelizmente, no dia de hoje eu não pude comparecer ao evento e agradeço muitíssimo aos professores Renato Saboia e Eleonora Dossi, que gentilmente aceitaram me representar. Tem sido um desafio a coordenação desse projeto, mas os resultados promissores nos fazem prosseguir. Espero que possamos nos encontrar em breve e desejo a todos um ótimo evento.
Caiu.
Olá. Não, passou o vídeo.
Não, ele é maior do que isso, ele caiu no meio.
Então, deixa eu tentar de novo aqui. Mas era o vídeo certo?
Sim, era o vídeo certo, só que ele foi interrompido.
Então, vou tentar de novo aqui. Só um minuto que eu estou tentando copiar ele de volta.
Nos encontrar em breve e desejo a todos um ótimo evento.
Olá, Eu sou o Dr. Adalberto Lopes, especialista em geoprocessamento, e desde novembro de 2019 estou realizando estágio de pós-doutorado pelo PRINT. Como uma das experiências no exterior que me capacitaram para esse estágio, fui pesquisador visitante na Queen's University of Belfast, no Reino Unido, na School of Nature and Built Environment. Com a supervisão do professor Dr. Cassiano Heck, estou desenvolvendo um índice de livability para a cidade de Florianópolis, como parte do projeto Envelhecimento Saudável e Mobilidade Urbana. Ainda tenho desenvolvido algumas atividades em conjunto com os membros do grupo de estudo e pesquisa em ambiente urbano e saúde, e colaboração com pesquisadores dos programas de pós-graduação em Educação Física, Saúde Coletiva e Arquitetura e Urbanismo da UFSC. Grato pela oportunidade, desejo a todos um excelente evento.
Olá, meu nome é Soraya, eu sou aluna do programa de pós-graduação em saúde coletiva. Atualmente estou fazendo doutorado sanduíche aqui na Universidade de Queensland, em Brisbane, na Austrália. Eu tenho bolsa pelo programa CAPES PRINT no projeto Envelhecimento Saudável e Mobilidade Urbana. O tema da minha tese é a teleconsulta médica e está sendo uma oportunidade incrível conviver com pesquisadores da área que já pesquisam esse tema há muito tempo, pois a teleconsulta ela já é parte dos modelos de cuidado e do sistema de saúde aqui na Austrália há muitos anos. A gente tem um artigo em parceria com o meu orientador aqui da Austrália, o professor Len Gray, e a minha orientadora no Brasil, a professora Maria Cristina Calvo. A gente já tem um artigo, então, submetido, e existem 2 outros artigos dos quais eu faço parte, pelo grupo de pesquisa que é voltado principalmente para a inserção das ferramentas de saúde digital nos modelos de cuidado, com uma atenção voltada para as doenças crônicas, especificamente para o diabetes tipo 2. Eu acredito que essa experiência, para mim, profissionalmente, está sendo fantástica, né? Ampliando os horizontes. Eu acho que é muito importante que os pesquisadores brasileiros tenham essa oportunidade de passar um tempo fora, ver como a pesquisa acontece em outros países, onde existe mais investimento, mais recursos, valorização do trabalho, né, do pesquisador e da pesquisa em si. Então está sendo uma oportunidade fantástica.
Olá pessoal, eu me chamo Larissa Weber, sou doutoranda do programa de pós-graduação em saúde coletiva, na linha de avaliação em saúde. Estou há 1 mês em Lisboa, realizando o doutorado sanduíche na Universidade Nova, sob orientação da professora Isabel Craveiro, dentro do subprojeto Envelhecimento Saudável e Mobilidade Urbana. Aproveito para agradecer a oportunidade e desejo a vocês um excelente evento. Um abraço.
Olá, meu nome é Andréa, eu faço parte do projeto PRINT Mobilidade e Envelhecimento Saudável, onde a gente tem os cursos da saúde coletiva, da arquitetura e da educação física. Eu faço parte da pós-graduação da arquitetura e eu tive a oportunidade de fazer uma missão acadêmica para a Holanda, visitando a Universidade de Groningen, onde eu pude conversar com o professor Birros e a gente conseguiu com que eles aceitassem 2 doutorandos para fazer uma dupla diplomação entre os nossos programas, sem as taxas a serem pagas, né? Com um trâmite de 6 meses para que a gente pudesse fazer todos esses encaminhamentos, além de eles identificarem também um projeto para a gente conseguir fazer os pós-doutoramento júniores, né? A possibilidade de virem para trabalhar junto conosco em congressos internacionais, estruturação de pesquisas, de trabalhos, porque na Holanda o banco de dados que eles têm em termos de informações epidemiológicas, genéticas, do grupo não só etário de idosos, mas do grupo populacional é muito grande, muito enriquecedor para a gente fazer essas pesquisas em conjunto. Tive a oportunidade também de ir a Portugal apresentar 2 trabalhos no Congresso sobre Planejamento Internacional sobre as Cidades, pensando esses planejamentos de cidade, principalmente apresentando a parte da caminhabilidade, e também na Alemanha, no 9º Simpósio Brasil-Alemanha sobre o Desenvolvimento Sustentável, onde eu pude apresentar 2 trabalhos também correlacionados com o envelhecimento populacional e a mobilidade, pensando em cidades saudáveis, pensando em cidades ativas. Então, as missões, elas são excelentes para que a gente pudesse desenvolver melhores parcerias e as melhores redes, né? Isso aconteceu já em 2019 e esperamos que continue a prosperar nos próximos anos.
Sou professora Lisandra Vergara, do Pós-Arc, e faço parte do PRINT de Envelhecimento Saudável e Mobilidade Urbana. Em dezembro de 2019, eu tive a oportunidade de fazer uma missão acadêmica para a Universidade de Illinois, em Champaign-Urbana, Estados Unidos, onde eu conheci e firmei parceria com o Departamento de Kinesiologia e Community Health, onde a minha supervisora, Wendy Rogers, está desenvolvendo um laboratório que é a Life Home para realizar pesquisas com idosos. Então, a área que atuo dentro da ergonomia, ela envolve a questão das tecnologias assistivas para idosos. E nesta visita técnica, tivemos a oportunidade de viajar para conhecer o Thrive Center, que foi um showroom de exposição de tecnologias para idosos, que foi realizado em Louisville, né? Foi bastante interessante esse contato, criou oportunidades para o pós-doc. Então, em março de 2021, estarei indo fazer o meu pós-doc, né, como professor visitante nessa universidade, e também tive a oportunidade de abrir uma bolsa de doutorado sanduíche pelo PRINT de uma aluna de doutorado minha do pós-arc. Acredito que o PRINT está sendo uma grande oportunidade para nós pesquisadores e principalmente para os alunos fazer esse intercâmbio e poder criar e produzir mais em conjunto com os nossos parceiros internacionais.
Olá, eu sou o professor Renato Saboia, estou atualmente como professor visitante na Oxford Brookes University, no Reino Unido, sob supervisão do professor Ramin Keivani. Infelizmente, devido à pandemia, a universidade encerrou suas atividades presenciais em março desse ano, mas antes disso, nos 2 meses que eu passei lá, eu tive a oportunidade de conhecer e de vivenciar várias experiências muito interessantes. Uma delas, participar da disciplina de escrita acadêmica, nessa parte que é super importante no trabalho de qualquer pesquisador. Uma outra experiência bem interessante que eu participei foi a de reuniões com professores de diversos grupos e faculdades da universidade, em que eles destrinchavam editais de financiamento que tinham sido lançados pelo governo e tentavam verificar se eles conseguiriam montar propostas em conjunto. E, por último, uma outra experiência foi a de uma reunião em conjunto com professores da Universidade de Oxford, de um projeto de pesquisa chamado Peak Urban, em que professores da Oxford Brookes e professores da Universidade de Oxford apresentavam as suas pesquisas, os temas em que eles estavam trabalhando no momento, em busca de temas em comum que pudessem depois suscitar novas pesquisas em conjunto, em colaboração, entre grupos de pesquisa dessas 2 universidades. Então, poder verificar como é que eles fazem esse tipo de coisa. Atualmente, então, eu estou trabalhando numa pesquisa em cima dos dados do Floripa, que é um questionário bastante extenso, feito com idosos em Florianópolis, feito por uma equipe coordenada pela professora Eleonora Dorsey, que aplicou esse questionário a 1.705 idosos, e que agora eu estou então limpando esses dados e adequando esses dados à minha pesquisa, mais especificamente, que é tentar entender como os idosos percebem a diversidade de uso do solo e como que essa percepção e a própria diversidade de uso do solo podem influenciar na decisão desses idosos de caminhar ou de não caminhar pelo seu bairro. Então agora eu estou aplicando alguns scripts, algumas técnicas de programação para limpar bem esses dados, para reformatar, para verificar se existe alguma inconsistência, e estou produzindo as colunas com as variáveis que vão ser relevantes para mim nessa pesquisa, para a gente poder entender como é que esse fenômeno funciona e como é que a gente pode pensar, talvez, políticas públicas que sejam um incentivo à caminhabilidade nesse grupo etário.
Diego.
Alexandre, por favor, me coloque como apresentador.
Já estás como apresentador, Diego.
Obrigado.
Diego, você está como apresentador, então. Em breve já deviam ser arquivos carregados. Obrigado, Diego.
Legal, acho que agora sim. Todos veem a apresentação, né?
Sim, tá tudo certo.
Hello, everyone. I'm a coordinator of the project Diagnosis Intervention for Health Lifestyle. This project is a partnership between two graduate programs at UFSC, Physical Education and Public Health. The main proposal of this project is to study different actions for diagnosis and interventions for a healthy lifestyle. So we have the focus on healthy lifestyle, but we study the analysis of contextual aspects of determinants in human health. We use as a model, theoretical model of socio-ecological model to understand healthy lifestyle. So we study different contexts of the health, of the human health. For that, we have a team with 15 professors who are involved in different research to find better ways of diagnosis interventions for a healthy lifestyle. Of course, in addition to these professors, we have the entire group of students and other professors from the both graduate programs involved. We have a partnership with 22 universities from other countries across the world. A good example is the partnership with Professor Andiara, who spoke to us today, but also we have a partnership with universities in Germany, Australia, Belgium, Canada, US, France, Italy, Norway, and UK. This map shows the partnership that we have with across the world. We have different actions to internalizations of our project. We had some important actions in the early years. For example, we sent 5 PhD students for an internship period in US, Canada, and Australia. We sent 2 professors of our project as a visiting scholar to Canada and Australia, and we did technical visits to different labs around the world. We received a professor from Canada as a visiting scholar, and we received other professors for technical visits. Another good example of your project was we received the UK students to postdoc with us. That was interesting actions of your project. We have, for sure, important challenges for the coming years. Special attention will be paid to the context of the COVID-19 pandemic and the effects of the situation on people's lifestyle. In addition, we aim to promote a high-level training of human resources with the following focus: effects of physical activity on physical and mental health indicators, effects of sedentary behavior on physical and mental health indicators also. In addition, we study community health intervention models for a healthy lifestyle. Also, public policies to promote a healthy lifestyle, we all focus. And also, health education programs to promote a healthy lifestyle will be all focus in the next years. This was all actions in this project. We have no video, so thank you very much for your attention. The next one, João.
Yes. Me ouvem?
Sim, João.
Ok. Agora eu preciso entrar com a minha apresentação, né? Eu já estou como apresentador, não?
Agora sim, João.
Acabei de me colocar.
Certo. E agora eu tenho que achar onde é que está o mais aqui. É o que está à esquerda aqui? Esse mais que está à esquerda?
É, um botão azul à esquerda embaixo da área escura.
Certo. Eu vou abrir compartilhar um vídeo externo, carregar uma apresentação. Aqui, agora eu preciso... para carregar tem que trazer aqui, não dá para entrar no direto... Tem que abrir aqui, então, só um momentinho, gente. Está carregando, será?
Sim, depois de selecionar o arquivo, tem que dar o enviar, ele demora um pouquinho para carregar, porque ele converte.
Tá certo.
Você ainda está na tela toda branca?
Não, não está carregando. Está dizendo que está carregando, está dizendo que 30%, né, foi carregado.
Ok.
Está aparecendo aqui.
Daqui a pouco deve aparecer para todos, então.
Obrigado. Porque eu acho que, como tem vídeos, né, porque eu anexei o vídeo dos alunos de um professor que fez, que saiu em estágio, né, de professor visitante no exterior, está anexado. Está bem lento. Eu não sabia que o PowerPoint, eu não me lembrava que o PowerPoint teria que fazer essa conversão, porque eu praticamente não uso essa plataforma. E está dizendo que é muito grande, que é para dividir o conteúdo. Então, se você puder, botei esse link, por favor, coloque esse link, e a primeira parte dele está sem voz, aí eu falo, em cima da primeira parte, e a segunda parte entra o vídeo. Talvez seja a melhor saída. Você acha que pode ser assim, Alexandre?
Está carregando, deve parar de carregar.
Não, é porque falou que tinha que dividir em dois. O vídeo era muito grande. Então é melhor você... Eu coloquei um vídeo externo, eu coloquei um link do YouTube, eu subi no YouTube agora.
Esse vídeo, então, do link.
É um link. E daí o vídeo vai estar nesse link também, só que as três primeiras... No início, eu vou falar em cima do vídeo. Tem problema?
Não, a gente baixa o som do vídeo.
Eu falo em cima do vídeo, porque pelo PowerPoint não vai dar.
Vídeo.
Aí, por favor, use esse vídeo aqui, eu já subi ali no YouTube. Em qual hora que eu vi que ia dar problema, né? Você me diz a hora que tiver já...
Está carregando, tá já passando o vídeo, está em play.
Certo. Bom, boa tarde a todos. Eu sou... O pessoal está dizendo para falar em português? Fala em português ou fala em inglês?
Acho que ficou agora só os em português na sala.
Bom. Então, volta o vídeo para mim para o início aí, senão não vai passar aqui, eu não vou conseguir falar, desculpe. Bom, pessoal, boa tarde a todos. Eu sou o João Borges Laurindo, né? Sou coordenador do projeto, então, processamento de alimentos de origem vegetal e seu impacto na saúde. Nesse projeto participam 2 programas de pós-graduação, o programa de pós-graduação em engenharia de alimentos, o programa de pós-graduação em food science. Infelizmente, saiu o programa de nutrição por questão das notas da CAPES. Nós temos tido colaboração efetiva com a Universidade da Califórnia, Universidade de Cornell, com a École Nationale Vétérinaire de Nantes, com a McMaster University, com a Universidade Wageningen e com a Universidade de Parma, na Itália. São colaborações que estão ocorrendo, né, com alunos e professores, e estamos recebendo professores de lá também. Os principais projetos, então, poxa, para ficar em inglês tem que trazer para o português, né? Aplicação de processos enzimáticos verdes, né, para obtenção de alimentos funcionais, a partir de plantas de oleaginosas de maneira geral, né, com destaque aí para a castanha do Brasil, o efeito de novas tecnologias na qualidade de proteínas derivadas de plantas, né, principalmente de resíduos das indústrias produtoras de óleos, incluindo aí a formulação de análogos de carne, né? E também temos desenvolvido novas estratégias para processamento de micro e macroalgas, desde a produção até o processamento. Obviamente que a produção é em cooperação com a aquicultura, o professor Justino, em colaboração com a professora Leila. O cereal sourdough fermentation, né, tem sido estudada para a redução dos FODMAPs. Volta um pouquinho, porque tá só um pouquinho para mim aí. Atrasa um pouquinho o vídeo aí, só por favor, senão eu não consigo... Eu não tenho controle nenhum, é problema disso aí. Só 10 segundos. Atrás só para eu... Isso. E todos eles, então, relacionados à questão da saúde, inclusive com questão da digestão in vitro, que a gente tem trabalhado, né? e estudar da digestão in vitro. E uma outra parte do projeto seria, então, o desenvolvimento de equipamentos de pequena escala, que nós estamos chamando o projeto Fábrica de Fábricas, né, com financiamento do MCTI, saiu hoje no Diário Oficial. Então, nós estamos construindo mini fábricas instaladas dentro de containers para desidratação da cadeia de açaí com cupuaçu, para agregação de valor nessa cadeia. Nós vamos instalar essas fábricas em várias comunidades tradicionais, inclusive uma comunidade de mulheres indígenas, né, nos Solimões, né, e uma comunidade de mulheres da Ticuna. E o estudo utilizando processos, né, de baixa pegada de carbono, e também nós vamos acessar, né, e determinar o ciclo de vida dos produtos. Então, essa é a ideia, né, de gerais, assim, projetos, assim, do curso. Agora aqui eu acho que vou ter que aumentar o volume um pouquinho.
Only is a recognized school in the agri-food sector in France, and the lab I work in is large, super organized, with many equipment for processing and analyzing food. I work with food drying, so I ran experiments in Brazil, and now I'm finishing my test here in France. I prepare starch solutions with different concentrations of sugar to simulate a food suspension, and then I dry them in an equipment called cascade drying, analyzing also the dry product. This is one of the equipments that I work on. This is a small-scale cascade drying, heated by electrical resistances. I prepare starch solutions with sugar on it. This is another cascade drying, but with circulation of heated water, and we can do the spreading of product here. With the equipment I'm using right now, we can quickly cool the product down to a certain temperature. This partnership between universities in Brazil and in France was extremely important to me. It made all the difference for my professional and personal growth. I was able to know another work environment in another country, and at the same time I realized how much we have resources and technical capacity in Brazil to do good research. So this experience enriched me a lot, because it added knowledge to the level of research. I was able to improve my thesis while working together with my supervisors in Brazil here in France, and I was able to learn a new language, a new culture. So those knowledges will certainly be of great value to my professional path.
Hello everyone, my name is Ana Salvador. I'm a PhD student in the Department of Food Engineering at the Federal University of Santa Catarina. My advisor in Brazil is Professor Bruno Carcioffi. Since March of this year, I'm doing my internship at Cornell University in the United States. My mentor here is Professor Ashin Datta. Professor Datta is well known for his work in modeling heat and mass transfer in biological systems. He published many papers in the field, books, and here at Cornell University, that is one of the best universities in the world, he is responsible for 2 courses. He's also in charge of a lab group here with students from different fields. For example, mechanical engineering, chemical engineering, agriculture, medicine. His students came from different parts of the world: India, Thailand, Italy. He also has students from the United States. Besides all that, the lab is equipped with computers of high performance to help in our simulations. All this exchange of cultures, knowledge, is helping me a lot in my PhD. My PhD is the modeling and simulation of microwave vacuum drying of fruits and vegetables, and is strongly related with their work here. I am thankful for all this knowledge that we are sharing, for all the people that I knew here, for my scholarship, and I hope that this can bring more knowledge, information to our lab in Brazil too. Thank you.
Hi everyone. My name is Giustino Tribuzzi. I'm a professor at the Food Science and Technology Department and at the graduate program of Food Science at UFSC. And I'm here to talk to you briefly about my stay as a visitor professor at the University of California, Davis, with a scholarship provided by the PRINT-UFSC program. My stay was of about 5 months, and I joined the Food Engineering Research Group under the supervision of Dr. Gail Borchhorst. The researchers of this group are focused on the in vitro digestibility of food with dynamic models. And my project was about the study of the effect of different processing technology, such as drying, roasting, milling, on the in vitro digestibility of chickpeas. The in vitro digestion was performed in this device here presented in this video that is called human gastric simulator, that simulates a human stomach with peristalsis and periodic gastric emptying. With this system, one can study the biochemical and the physical modification of food during digestion and relate this phenomena with satiety and with glycemic response, for example. The studies are very multidisciplinary and involve different fields such as food science, food engineering, human nutrition. It was a great opportunity for me to deeply understand the dynamic that control the in vitro digestion, and I'm sure that this knowledge will allow our research group here at UFSC to give our contribution in this research field. Further, the internationalization experience improved the relation between both research group here at UFSC and the UCD, allowing future students and professor exchanges and joint research project. I would like to thank UFSC, UCD, and the program PRINT-CAPES for this amazing opportunity.
Era isso, professor?
Ele está mudo lá.
Sim. Finalizei. Era essa a apresentação. Obrigado. Desculpa aí um pouquinho de confusão aí. Mas é isso. Obrigado.
Obrigado, João. Agora é a apresentação do professor Edmundo. O professor Edmundo mandou um vídeo para ser passado, né, que representa o projeto dele, Comunicação Celular e Biologia de Sistemas, cujo PPG líder é Biotecnologia e Biociências.
Good morning. I'm here to present you our project entitled Cellular Communication and Systems Biology. Communication between the cells of an organism, between single-celled organisms, or between them and the host is essential for the maintenance of life. Our project is focused on fundamental aspects in the biological sciences, being one of the bases for a broader understanding of life sciences. Understanding the mechanisms involved on different levels of intra- and interspecific cellular communication and interaction is vital. This knowledge, mainly in the basic areas, is critical for the technological development of the most different areas of the productive sectors within Brazil and abroad, focused in human health, animal health, and the environment. Given the complexity of such tasks, cooperation between national and international research groups is crucial, notably with regards to exchange of staff, PhD, and postdocs, and also generation of knowledge in partnership. In Brazil,
Most scientific knowledge is generated from within the public universities, and thus international collaboration becomes mandatory in order to achieve worldwide prominence in science, technology, and innovation. The present project, funded by CAPES, has originated from within the UFSC Center for the Biological Sciences and aims the promotion of collaborative research, science, and contribution to the national and international relevance of our institution. The aim of our project is at the heart of the world interest to understand how cellular interactions occur within the species, allowing the understanding of biological phenomena such as human and animal infection by microorganisms, interaction of tumor cells with normal cells, and cellular interaction in chronic degenerative processes, among others. By congregating 20 Brazilian researchers, 6 post-graduation programs, and 9 international collaborators from 8 partner institutions, the project has so far succeeded to promote science and training of human resources.
Agora a professora Ariane, do PPG Bioquímica.
Ok, a professora Ariane já está como apresentadora.
Estou carregando a apresentação. Olá, pessoal, boa tarde a todos. A mesma coisa que aconteceu com o professor João, que eu tenho que dividir. Então, eu só vou apagar os vídeos e na hora eu peço para o Alexandre disponibilizar o vídeo, pode ser?
Pode ser.
Deixa eu só apagar daqui. Continua dizendo que é muito grande. O que pode ser isso, Alexandre?
Às vezes a apresentação tem muitas imagens, muitas páginas, talvez salvar em PDF, teria que preparar para o arquivo ficar menor.
Tá, vou colocar PDF, então. Será que é porque eu deixei só o link do YouTube? Tem que tirar também o link?
Não vou saber ele confirmar agora, professora.
Tá. Vamos ver se o PDF carrega agora. Acho que agora foi.
Ok, aguardemos.
Pelo menos agora ele diz que está processando.
deve dar.
Bom, pessoal, mas enquanto ele vai carregando o vídeo, acho que eu já vou falando, porque já estamos no adiantado da hora. Bom, o meu nome é Ariane Zamoneri Pacheco de Souza, e eu sou coordenadora do subprojeto coordenado pelo Programa de Pós-Graduação em Bioquímica, e desse projeto também fazem parte o Programa de Pós-Graduação em Neurociências e o Programa de Pós-Graduação em Farmacologia da UFSC. Esse projeto, ele reúne pesquisadores desses 3 programas, além de pesquisadores de diversos países, de universidades de diversos países, como Estados Unidos, Espanha, França, Portugal, Inglaterra, Canadá, Austrália. Temos pesquisadores de universidades distribuídas em vários países. Eu não sei se talvez, Alexandre, o vídeo seria o mais rápido do que esperar abrir, porque bom, faltam 3 páginas ainda.
Não sei, se eu passar o vídeo agora vai parar de carregar.
Então espera terminar. Desculpem, pessoal. Foi. Bom, então, como eu já falei para vocês, eu sou coordenadora desse projeto que abrange 2 outros programas de pós-graduação, e nosso projeto é intitulado Molecular and Therapeutic Targets in Metabolic and Degenerative Diseases. O nosso projeto, então, integra pesquisadores interessados em compreender os mecanismos celulares e moleculares envolvidos no desenvolvimento de diversas doenças crônicas. O nosso time internacional, os pesquisadores internacionais, estão distribuídos, então, em várias universidades em torno do globo: Universidade de Leuven, Albert Einstein College of Medicine, Universidade do Porto, Universidade de Barcelona, Harvard Medical School, Universidade de Montreal, University of Victoria, Instituto Karolinska, na Suécia, Universidade da Califórnia, Berkeley, University of Calgary, no Canadá, Universidade Miguel Hernández, na Espanha, a Macquarie University, na Austrália, entre várias outras universidades que fazem parte das cooperações internacionais dos professores desses 3 programas de pós-graduação. Os pesquisadores, eles estão envolvidos na pesquisa científica e tecnológica, inovação, e também no ensino. Nosso objetivo principal é a formação de recursos humanos voltados principalmente para a ciência básica, né, o que nós entendemos que é fundamental para o desenvolvimento socioeconômico de qualquer país. Dentre as ações já desenvolvidas de internacionalização por esses programas, eu vou salientar algumas atividades, principalmente nesses últimos 2 anos. Em 2019, nós tivemos a visita como professor visitante do professor Romain Grinamar, da Université de Caen. A Universidade de Caen tem sido uma parceira internacional bastante relevante nos projetos do Programa de Pós-Graduação em Bioquímica. Nós tivemos em 2018 a defesa de uma tese de doutorado em cotutela, da aluna, agora doutora Renata Gonçalves, onde nós recebemos a visita para participar como banca da professora Hélène Lelong. Atualmente, nós temos a aluna Emily Batista, que está na Université de Caen, desenvolvendo o seu doutorado sanduíche com uma bolsa da universidade, uma bolsa do governo francês, e também está fazendo uma tese de doutorado em cotutela, pelo programa de pós-graduação em bioquímica. Outras cotutelas já foram realizadas também pelo programa de pós-graduação em farmácia com essa universidade. Saliento também a participação no nosso projeto do professor Marco Alves, que está aqui hoje, participou dessa nossa atividade, e do professor Pedro Fontes Oliveira, que é membro do nosso subprojeto CAPES PRINT, tanto do subprojeto coordenado pelo PPG em Bioquímica, quanto um dos projetos coordenados pelo PPG em Farmácia e pelo PPG em Biotecnologia. Nesse ano, os 2 professores já participaram de atividades vinculadas a ações de internacionalização nos seminários que nós realizamos no programa de pós-graduação em Bioquímica, e convidamos outros programas para participar, que é o projeto Frontiers of Medical and Biological Science, Quarantine Edition. Então, semanalmente, nós temos nos reunido para discutir ciência, cada um em sua casa, e o professor Marco e o professor Pedro já participaram dessas atividades nesse ano de 2020. Ano passado, nós realizamos o primeiro ciclo de seminários internacionais em Bioquímica, Ciências Médicas e Farmacologia, onde nós recebemos, entre outros pesquisadores, o professor Sebastian Talbot, da Universidade de Montreal, que já nos visitou por diversas vezes, e apresentou seminários, participou de cursos, palestras, e de vários eventos, discussões científicas, visitas no hospital universitário. Então, participou de diversas atividades já vinculadas à internacionalização da UFSC, tanto com professores da farmacologia, da bioquímica e das ciências médicas. Nós temos uma aluna, atualmente, a Tuane Eichwald, que está na Universidade de Montreal, sob supervisão do professor Sebastian Talbot, e ela está realizando o doutorado sanduíche, que será em cotutela também, com dupla titulação. E aí, Alexandre, eu não sei se tem como, se eu clicar aqui, não vai exibir o YouTube, né? Dá para exibir o vídeo da Tuane sem sair da apresentação ou não?
Não vai sair da apresentação.
Mas aí, como já está carregado, vai abrir facilmente depois, ou não?
Suponho que sim.
Tá. Então, se você puder colocar o vídeo que a Tuane preparou para nós.
Tá, então eu vou ter que me tornar apresentador e passar o vídeo.
Deixa que colocamos os vídeos no final, então, para não sair, tá? Ficar mais prático, né?
Ok, ele aguardo.
Tá, então, ao final, a gente pode exibir para vocês um vídeo da Tuane apresentando um pouquinho sobre o que ela tem feito na Universidade de Montreal, no Canadá. Acho que aqui deu, um deles deu algum probleminha aqui. Nesse ano de 2020, um pouquinho antes de começarmos o distanciamento social devido à pandemia, bem no início de março, nós recebemos novamente o professor Sebastian Talbot, que está aqui, e também o professor Andrei Stroszczynski, da Universidade de Pittsburgh, e ele também tem atividades, então, na Universidade em Montevideo, onde nós realizamos, eles ministraram disciplinas para os programas de pós-graduação em Bioquímica, Ciências Médicas e Farmacologia. Estava aberto para vários programas de pós-graduação, então participaram de outros programas do CCB e do CCS, de atividades vinculadas à visita desses 2 professores. Programa de Neurociências também, temos aqui professores de outros programas participando. Então, no início de março, eles viajaram exatamente no final de semana em que começou o distanciamento social e a UFSC, né, fechou. Então, tivemos uma atividade agora em 2020. Aqui depois, então, ao final, a gente exibe o vídeo do professor Andrés, também comentando sobre a participação dele em atividades vinculadas ao programa CAPES PRINT. Professores do programa, então, também tem uma parceria bastante longa com a Universidade de Barcelona, o professor Francisco Ciruela, que concluíram uma tese de doutorado em parceria com o professor Francisco Ciruela, agora em setembro, do aluno Caio Massari Leite, do programa de pós-graduação em Bioquímica. Ele já recebeu vários estudantes aqui da UFSC, e ano passado também nos visitou em abril, para participar, então, de um evento que foi organizado em parceria com professores do pós-graduação em Farmacologia, Bioquímica e Neurociências. Nós recebemos o professor Francisco Ciruela. Aqui é uma egressa do PPG em Farmácia, a Patrícia, que esteve na Universidade do Porto, então, sob supervisão do professor Pedro Fontes Oliveira, que também estava aqui até há pouco participando, então, que trabalham em colaboração ele e o professor Marco Alves. Aqui nós temos a visita do professor, relatando a visita do professor Michael Aschner, que também já recebeu inúmeros estudantes brasileiros da UFSC e de outras instituições do Brasil, da Albert Einstein College of Medicine, do Departamento de Farmacologia Molecular. Então, aqui nessa visita, nós temos aqui o professor Marcelo Farina, o professor Rodrigo, da Bioquímica, o professor João Batista, da Federal de Santa Maria, o Félix também, de Santa Maria, e aqui o professor Michael Aschner, num evento sobre bioquímica toxicológica, organizado pelo professor Marcelo Farina. Aqui, então, ano passado, nós recebemos o professor Rodrigo Cunha, da Universidade de Coimbra, em Portugal, e o professor Francisco Ciruela, como eu já mencionei para vocês, para participar do meeting do Clube Brasileiro de Purinas, que aconteceu em 2019 em Florianópolis, e fizeram parte do comitê organizador local, então, professores do programa de pós-graduação em Bioquímica, Neurociências e Farmacologia. Aqui, então, depois eu vou colocar um vídeo da professora Alexandra Latini, que vocês vão ver ao final. A professora Alexandra Latini, no momento, está nos Estados Unidos, na Universidade da Califórnia, em Berkeley, e é orientadora, então, da Atuane, que também vocês verão no vídeo depois, e ela está, então, como professor visitante no exterior. Ela foi com o projeto vinculado ao projeto liderado pelo professor Edmundo, e como ela é docente do programa de pós-graduação em Bioquímica, ela gravou um vídeo aqui para nós, então, para falarmos um pouquinho sobre essa internacionalização, esses projetos de internacionalização que temos no CCB. Tá? Então, mais informações sobre o projeto de internacionalização liderado pelo Programa de Pós-Graduação em Bioquímica estão disponíveis no endereço eletrônico do programa, no endereço eletrônico da ProPG, e também vocês podem consultar, então, os e-mails da internacionalização da ProPG, pelo SIM, ou também o meu e-mail. Agora, então, eu gostaria de exibir os vídeos, agradeço a participação e a atenção de todos vocês, e pediria para o Alexandre para colocar, então, nós temos 4 vídeos, né, o do programa, e mais os vídeos dos alunos e do professor. Obrigada.
Estou começando com o que está como Tuane, correto?
Ok, obrigada.
Hello everyone, my name is Tiana. I am a PhD student in the Biochemistry Department at the Federal University of Santa Catarina under the supervision of Professor Alexandra Latini. Last year, I participated in a call for international scholarship from CAPES Print. I was selected, and since November 2019, I am here in the University of Montreal in the Sebastian Talbot Lab. And I would like to share with you what I have been developing and learning here and how much it has had a positive impact in my personal and professional life.
Methodologies such as modeling diseases in genetically modified animals to create mice whose neurons are ablated, silenced, fluorescent, activity-dependent, or light-sensitive reporters are developed here. After the model diseases, we collect their neuronal and immune cells to access their individual properties and understand how they interact with each other. Using the calcium imaging, we are able to characterize the neuronal responses to different stimuli, including the signals produced by the cells of the immune system. Another methodology developed here is the flow cytometry, that allow us to characterize the expressions of inflammation markers by neuronal and immunological cells. In addition, due to pandemic, I had contact with bioinformatics, and even in the period and the university was closed, I was able to learn a new methodology. All this knowledge is also being materialized in 5 different papers, one of each has already been published, 2 are under review, and 2 are in the writing phase.
Ok, obrigada. Agora nós vamos ver o vídeo do professor André Strochansky, que esteve como professor visitante no Brasil no início de março desse ano.
Buenas tardes, mi nombre es Andrés Troschansky, yo soy profesor adjunto visitante del Departamento de Farmacología de la Universidad de Pittsburgh y además profesor adjunto del Departamento de Bioquímica de la Facultad de Medicina en Uruguay. Y quería comentarles un poco mi experiencia con el programa de internacionalización CAPES PRINT, en el cual me ha permitido a partir de este año trabajar y colaborar con la Universidad Federal de Santa Catarina y en particular con el LAVOX, dirigido por la doctora Alexandra Latini, la cual me ha llevado a participar este año en el dictado de aulas en la universidad a comienzos del 2020, así como realizar actividades de investigación en dicha universidad. Y a lo largo de todo este año hemos continuado trabajando y continuamos trabajando en esta área de experimentación, ampliando además la red de colaboradores extranjeros, incorporando a esta red a los doctores Cepeda, Ramiro Cepeda de la Universidad de Chile y al doctor Gilles Guillemín de la Universidad de Sydney, con lo cual, junto con la doctora Latini, hemos ampliado, como decía anteriormente, el horizonte de países y de grupos de investigación que están colaborando en esta red, gracias al programa CAPES PRINT. Actualmente nos encontramos realizando tareas relacionadas al proyecto, teniendo reuniones con estudiantes, reuniones de laboratorio, etcétera, en la cual se presentan resultados, se discuten seminarios, con lo cual la experiencia de este programa es altamente recomendable y me ha permitido insertarme en la colaboración con la Universidad de Santa Catarina y esperamos que esta colaboración continúe y se afiance aún más. Y bueno, muchas gracias.
No próximo vídeo, relato da professora Alexandra Latini sobre a experiência de internacionalização que ela tem realizado ultimamente.
Professor linked to the graduate program in biochemistry, and I have been coordinating for more than a decade the lab of bioenergetics and oxidative stress. The lab is dedicated to the study of the biology of tetrahydrobiopterin metabolism and mitochondrial energy production in physiological and pathological chronic conditions. Chronic disease is one of the topics linked to the CAPES PRINT sub-project entitled Molecular and Therapeutic Targets in Metabolic and Degenerative Diseases. This project is coordinated by Professor Ariane Zamoner Pacheco de Souza. CAPES PRINT has allowed us to initiate new collaborations and to intensify established ties. For example, LAVOX organized the first scientific meeting CAPES PRINT from UFSC. The meeting was composed of a school on electron microscopy, a school on nociception, an informal meeting to discuss international collaborations with professors from Canada, Argentina, and Chile, a mini international symposium on degenerative diseases, and finally a poster presentation by 5th grade students from a local school. Another example of the activities supported by CAPES PRINT was an international mini symposium with the visits of professors Andrés Troschansky from University of Pittsburgh and Sebastian Talbot from University of Montreal. This meeting allowed the initiation of new collaborations. Today, 4 groups from Uruguay, Chile, Australia, and Brazil are working together to understand the molecular mechanism underlying the neurophysiopathology of COVID-19. If you want to know more, please visit our website.
E, por fim, o vídeo de divulgação do nosso projeto. Já peço desculpas que estamos nos alongando, mas é bem curtinho, são 2 minutinhos e pouquinho. Obrigada. Hello, my name is Ariane Zamoner Pacheco de Souza, and I am the coordinator of the CAPES PRINT project entitled molecular and therapeutic targets in metabolic and degenerative diseases. Our project brings together scientists from Federal University of Santa Catarina and researchers from countries from around the globe interested in understanding the molecular mechanisms favoring the development of chronic diseases. The Brazilian team is composed by researchers linked to 3 graduate programs from UFSC, namely biochemistry, pharmacology, and neuroscience. These graduate courses offer a long-standing tradition of excellence in research as well as in master and PhD student training. The international team is composed by leading scientists committed to the study of the physiopathology of chronic diseases from United States, Belgium, France, Spain, Portugal, Switzerland, Sweden, Canada, and Australia. In addition to the deep commitment to scientific research and teaching, the principal investigators involved in this project understand the need to train highly qualified human resources for the generation of basic knowledge, which we understand as the fundamental basis for sustainable socioeconomic development of any country. The research topics covered by our project include the following: cancer, infertility, obesity and associated diseases, neuroscience and neurodegenerative diseases, redox process in biomedicine, toxicological sciences, cell signaling, drug discovery. The multidisciplinarity of this project allows brainstorming and product development research to tap into creative thinking and to test new concepts to contribute for innovation in the treatment of chronic disease. Visit our website and contact us to know more about CAPES PRINT-UFSC. Muito obrigada a todos. Eu passo para a professora Jussara, professor Diego.
Professora Jussara, apresentando o PPG Enfermagem.
Ok, obrigada, Diego. Boa tarde a todos e todas. Deixa eu só colocar aqui a apresentação.
Boa tarde.
Boa tarde. Eu não estou conseguindo colocar aqui, só um minuto.
Ok, aguardamos. Se quiser que eu vá passando algum dos vídeos.
Eu estava com o arquivo aberto, e aí na hora de colocar na apresentação, eu acabei perdendo o arquivo.
Lei de Murphy.
É verdade. E agora eu não estou encontrando nem nos acessos rápidos aqui. Mas eu penso que também é possível fazê-lo sem a apresentação, desculpem. Nós estamos finalizando as apresentações com o projeto de educação interprofissional, implementação de uma rede internacional de inovação e desenvolvimento do ensino superior. Fazem parte do projeto o programa de pós-graduação em enfermagem, como programa líder, e também os programas de pós-graduação em saúde coletiva e assistência farmacêutica. Nós temos desenvolvido parceria e atividades com as universidades de Nebraska, nos Estados Unidos, Toronto, no Canadá, com a Universidade de Lisboa, com a Universidade de Nottingham, no Reino Unido, e com a Kent University, também do Reino Unido. Durante o desenvolvimento destes 2 anos de programa, nós temos realizado atividades de doutorado sanduíche dos nossos estudantes e também a programação de professores visitantes que estiveram conosco nesse período e também professores dos programas envolvidos que participaram de atividades nas universidades parceiras. Nós realizamos, no início do projeto, uma atividade de lançamento, onde contamos com a presença do professor Michael Kalman, da Kent University, com atividades de workshops, conferências e desenvolvimento de disciplinas, bem como no seminário realizado ao final do primeiro ano, tivemos a participação da professora Claire Anderson, da Universidade de Nottingham, do professor Francisco Esteban e do professor José Luiz Medina, da Universidade de Barcelona, que eu não havia mencionado e que também é parceira do projeto, com a realização de um seminário e desenvolvimento de disciplinas, de workshops e de reuniões com os nossos estudantes, atividades que sempre ocorreram de modo aberto, com a participação dos estudantes e docentes de outros cursos do Centro de Ciências da Saúde e de outras universidades parceiras do programa de pós-graduação em enfermagem. Tivemos 4 estudantes que realizaram, ou ainda estão realizando, desenvolvendo bolsa de doutorado sanduíche. Tivemos bolsistas na Espanha, na Universidade de Barcelona, 2 bolsas de doutorado sanduíche e um pós-doc. Também estamos com uma aluna do doutorado de enfermagem que está concluindo agora em dezembro a bolsa de doutorado sanduíche nos Estados Unidos, na Universidade de Nebraska. Temos trabalhado também com o auxílio de uma colega colombiana que está realizando conosco atividades de professora visitante no Brasil, desde janeiro de 2020, a professora Maria Fernanda Vasques, que tem desenvolvido disciplinas, 2 disciplinas já sobre a temática do projeto, com atividades de discussões e simulação de educação interprofissional. ela também está trabalhando no desenvolvimento de pesquisas com os estudantes dos programas envolvidos e tem atuado fundamentalmente no desenvolvimento da temática e na parceria e no contato com as organizações internacionais que trabalham no âmbito da educação interprofissional. Temos a possibilidade de desenvolver agora, a partir de uma maior mobilidade por conta do controle da pandemia, temos a previsão de receber a visita do professor Ricardo Ayala, que é um colega chileno que atua na Universidade de Ghent, na Bélgica, também para fortalecermos as atividades no que diz respeito à intenção de sensibilizar o Centro de Ciências de Saúde para o desenvolvimento de práticas de formação, de educação interprofissional. Todas essas atividades têm sido desenvolvidas com ampla divulgação e com o convite sempre a que todos possam colocar as suas propostas, experiências e intenções em relação às atividades. Eu gostaria de, para encerrar essa participação, pedir se o Alexandre pode colocar os 2 vídeos que estão, os links do YouTube aqui na página. O Arthur fez a gentileza de colocá-los aqui. O primeiro vídeo é da Thaís Alves, da nossa aluna de doutorado, que está nos Estados Unidos nesse momento, e o outro da professora Maria Fernanda Vasques. Por favor, Alexandre.
Olá, meu nome é Thaís Alves, eu sou aluna de doutorado em enfermagem da UFSC. Eu sou aluna de doutorado em enfermagem da UFSC. Estou no 3º ano do doutorado. E esse ano de 2020 eu estou fazendo meu doutorado de sanduíche pelo projeto PRINT, no Colégio de Saúde Pública, Departamento de Promoção à Saúde, da University of Nebraska Medical Center, em Omaha, nos Estados Unidos. Minha bolsa é de 12 meses e eu cheguei aqui no mês de janeiro. Portanto, ficarei até o fim do ano. Eu estou estudando especificamente com o Laboratório de Pesquisa de Ciências da Disseminação e Implementação, o REAIM Framework, é o que eu vim aprofundar os conhecimentos aqui, e também tenho estudado outras teorias de disseminação e implementação para que eu possa aplicar na minha tese. Além das pesquisas relacionadas especificamente à minha tese, eu tenho participado de outros grandes projetos com os pesquisadores desse laboratório, então aproximando as 2 universidades, e tenho aprofundado diferentes estratégias metodológicas. Tive oportunidade de participar de estudos de validação e adaptação cultural de instrumentos, participei de pesquisas de métodos mistos na coleta e análise de dados, e também participei de ensaio clínico randomizado na parte de coleta e análise de dados. Eu acredito que essa experiência vai ser riquíssima, já tem sido riquíssima, não só para me ajudar a produzir uma tese muito melhor, né, uma tese que é voltada para o Sistema Único de Saúde, mas também no futuro como pesquisadora e como docente, que é a carreira que eu pretendo seguir, porque essa experiência do doutorado de sanduíche, ela nos aproxima de outras realidades, ela nos aproxima de outros contextos e permite com que a gente aprofunde os nossos conhecimentos, os nossos relacionamentos com outros pesquisadores. Então, certamente é uma oportunidade excelente que eu estou muito feliz de ter conseguido fazer. E os laços estão mais estreitos com os nossos laboratórios de pesquisa, e eu tenho certeza que essas produções seguirão sendo feitas em conjunto, mesmo após o término do meu doutorado de sanduíche.
Olá a todas e todos. Meu nome é Maria Fernanda Vasques, eu sou colombiana e neste momento sou bolsista jovem talento estrangeiro atuando no subprojeto PRINT, intitulado Implantação de uma Rede de Saúde Internacional para a Inovação e a Qualificação do Ensino Superior em Saúde. Esse projeto se desenvolve junto aos programas de pós-graduação em enfermagem e o programa de pós-graduação em saúde coletiva. Para mim, esta bolsa tem sido uma grande experiência, tanto profissional, acadêmica, quanto de pesquisa. Ainda com as dificuldades que todos nós temos experimentado esse ano, os encontros com os grupos de pesquisa que estão vinculados ao projeto, com os alunos das disciplinas que eu ministro também no marco do projeto, assim como as diversas palestras, atividades, webinars e cursos que tenho participado, têm permitido estabelecer parcerias novas, momentos de reflexão e de atuação importantes para o desenvolvimento do projeto. Acho que esse tipo de iniciativas são muito importantes de se manter no tempo para o avanço do conhecimento e o estabelecimento de vínculos entre os pesquisadores da região latino-americana. Vínculos que são, ao mesmo tempo, acadêmicos, mas também que são de afeto e de carinho. Agradeço pela oportunidade. Um abraço.
Com a palavra agora o Diego.
Obrigado, Alexandre. Só carregar a minha webcam. Acho que agora sim. Bom, pessoal, foi uma mesa diversificada, no sentido que foi bem extensa, mas bem profunda nos temas que foram abordados. Nesse sentido, a mesa temática da área de saúde humana, no âmbito do programa CAPES PRINT da UFSC, ela se encerra, né? E só uma breve retrospectiva da mesa, né, que essa área de saúde humana, no âmbito do programa CAPES PRINT na UFSC, envolve 11 programas de pós-graduação, entre programas líderes e programas parceiros, né, uma grande quantidade de programas que estão envolvidos, de 4 diferentes centros da nossa instituição, o Centro de Ciências da Saúde, o Centro de Ciências Biológicas, Centro de Desportos, Centro Tecnológico, docentes de diferentes departamentos, o que mostra que a área de saúde humana é uma área multidisciplinar e que envolve uma grande quantidade de pessoas. Se computarmos, né, por alto quantos docentes estão envolvidos nesses diferentes programas, a gente chega no quantitativo de aproximadamente 200 docentes da UFSC envolvidos no âmbito do programa CAPES PRINT na área de saúde humana, e de instituições parceiras, a gente computa mais de 50 instituições estrangeiras parceiras dos projetos no âmbito do programa CAPES PRINT. Nesse sentido, todos os coordenadores dos projetos no âmbito do programa CAPES PRINT na área de saúde humana agradecem a CAPES pelo suporte financeiro e apoio nessa jornada, agradecem à Pró-Reitoria de Pós-Graduação, agradecem à Universidade Federal de Santa Catarina e a todos os programas envolvidos na área de saúde humana. Um especial agradecimento aos colegas do LED UFSC por todo o suporte técnico durante essa jornada. Desejamos a todos que aproveitem. Amanhã é o próximo dia de evento. Espero que todos fiquem bem. Muito obrigado a todos. Nesse sentido, convido a professora Cristiane Derani, pró-reitora de pós-graduação da UFSC, para os últimos informes.
Muito obrigada, professor Diego. Eu gostaria de rapidamente encerrar esse longo dia que se iniciou às 9 da manhã e está terminando aqui às 7 da noite. Eu acho que isso é só uma amostra de quanto nós temos para falar, porque eu sei que todas as mesas tiveram ainda muito para dizer, mas estamos permitidos pelo tempo. Eu quero dizer que é muito importante esse evento, porque todos nós estamos tendo a oportunidade de nos conhecer. Então, a primeira mesa, internacionalização e interculturalidades na pandemia, com a temática 1, a segunda mesa sobre Nanostructured Ceramics with Enhanced Functionalities by Oculus Slurry Processing, uma mesa internacional muito, muito boa, muito interessante, de ponta, e agora, né, para finalizar, a mesa da temática saúde humana, que mostra o quanto nós trabalhamos com as preocupações e as soluções para aquilo que é as maiores necessidades da nossa sociedade contemporânea. Eu quero agradecer a todos que participaram durante esse dia e convidá-los para amanhã, às 9 horas, para seguirmos com nossa apresentação, mas um especial abraço ao nosso querido Alexandre Pinho e ao nosso querido Arthur, que ficaram aqui, né, desde as 9 da manhã, para poder auxiliar todos os professores e fazer desse evento o coroamento de 2 anos de serviços árduos, mas bastante gratificante. Uma boa noite a todas e a todos.
Obrigado, professora. O Laboratório de Educação Digital do Departamento de Engenharia do Conhecimento também agradece por poder dar o apoio ao projeto PRINT-CAPES-UFSC.